You are currently made of around 37 trillion cells. Every time you breathe, blink, or think, a massive city of living machines operates in the background to keep you alive. Yet every single one of those microscopic units originated from just one single cell. While early microscopic observations of cork revealed these honeycomb-like physical structures, modern science reveals they are actually dynamic machines, constantly processing the instructions that sustain life. Growth and healing rely on this mechanical reality. When you grow taller or your skin repairs a scraped knee, your body physically builds new tissue by commanding single cells to divide. To see exactly how that happens, we need to move past the outer membrane of a single tissue cell and travel into its center. Here we locate the nucleus, the control room storing the DNA that dictates how the organism operates. Understanding how a single microscopic event splits and replicates that genetic manual explains how complex life physically builds and rebuilds itself over an entire lifetime. The chronological process of one cell becoming two is called mitosis. It relies on an elegant mechanical sequence of steps to safely handle the genetic material inside the nucleus. Before a cell even attempts to divide, it spends a long period in a preparation stage called interphase. During this time, the cell simply lives, grows, and silently copies its entire DNA manual, ensuring there is enough genetic material to eventually supply two separate cells. When division begins in prophase, that loosely copied DNA suddenly packs itself into tightly bound, visible structures called chromosomes to prepare for transport. During metaphase, these newly formed chromosomes migrate inward to neatly line up directly down the vertical center of the cell. This meticulous, centralized alignment is an essential failsafe. It ensures that both future cells will receive the exact same genetic blueprint. The explosive action of anaphase follows, where the neatly aligned chromosome pairs are pulled apart to opposite ends of the cell. With the genetic materials safely separated, consider what must physically happen next to finish the creation of two new independent buildings. In telophase, a new protective nuclear membrane forms around each isolated set of DNA to secure them. Finally, the entire cell pinches down the middle to cleanly separate the two halves in a process called cytokinesis. This final physical separation transforms what was once a single, vulnerable cell into two fully independent units ready to start the cycle over. The ultimate outcome of this entire process is the creation of two perfectly identical daughter cells resting side by side. Having identical genetic instructions in both cells guarantees that the body functions consistently without losing vital biological data.